Well, good evening, everyone. We're going to go ahead and get started this evening. We're very excited for this inaugural Grand Rounds webinar series. Hello to everyone. I am Jeffrey Hayes, the ASRM Education Specialist and moderator for this webinar. Before beginning the webinar, I'd like to go over a few housekeeping items with everybody. To make sure we can cover all the content in the allotted time, everyone's line except the speakers will be muted. We will devote about 15 minutes at the end of the presentation to questions. Please feel free to type a question in the chat window at any time. I will then read as many selected questions as possible to the presenter during the allotted question and answer time. If for some reason you need to step away from our presentation this evening, please sign out and then sign back in on your return. After the webinar is done, if you would like to obtain the opportunity to get CME credit for this activity, please return to ASRM eLearn to take the post-test and, importantly, to complete the survey for your CME credit. You must complete the post-test questions successfully and complete the survey to receive credit, and then be able to print off your certificate. Our speaker today, or this evening, is Dr. Francesco DeMayo. Dr. DeMayo is a professor of molecular and cellular biology and pediatrics at the Baylor College of Medicine in Houston, Texas. We are so very excited for his talk today. So I will now let me go ahead. I'm going to unmute Dr. DeMayo, and I'm going to hand things over to him. If you would, sir, go ahead, and we are ready.
Okay, I'd like to thank ASRM for giving me this opportunity to present the Grand Rounds. I appreciate the fact that it's the eve of a 4th of July weekend, and I think there's probably some fireworks going off at the ASRM office right now with Andy's audio speakers. What I'm going to talk today about is the mechanism of action of steroid hormones, and I'm going to focus on it in endometrial biology using mouse models and primary human samples. So endometrial function is a real important healthcare concern. If you just look at infertility, about 7.3 million women U.S. Williams of reproductive age are infertile. And even if you look at natural mating, its efficiency is very low. About 30% of natural matings result in a pregnancy, and 75% of these pregnancies fail due to failures for the embryo to implant. And this is because the uterus regulates implantation by only allowing it during a specific window of implantation, where the uterus will allow the embryo to attach, invade, and support pregnancy. And S.K. Day, in a nice review in Nature Medicine, pointed out that problems with implantation don't only affect fertility per se, but have a lot of ripple effects on how pregnancy progresses. Problems with implantation have been tied to placental defects, preeclampsia, intrauterine growth retardation, spontaneous abortion, and obviously pregnancy loss and preterm birth. So understanding what the molecular mechanisms or what regulates endometrial function is critical. And since the uterus is the most sensitive hormones, the most sensitive organ to steroid hormone action, again, it's a perfect organ to study steroid hormone regulation. So what I'm going to talk to you today about, and this is a roadmap of my talk, is I'm going to give you an overview of the basic molecular biology of nuclear receptor signaling. I will then show our work in defining the role of progesterone receptor in the mouse uterus, and then identifying what progesterone receptor is doing in the mouse uterus, and then go specifically into looking at not only progesterone receptor, but how it's co-regulated to regulate uterine function. Then I'll talk about the epithelial-specific expression of progesterone receptor and its role, and then we'll go ahead and talk about how we can do some of these findings in the human endometrium. So we're going to start with the nuclear receptor signaling. And so this is the stick figure of what most of the nuclear receptor structure. The nuclear receptors are the largest family of transcription factors. They regulate gene expression. If you look, they consist of a ligand binding domain. So in the case of receptors that have identified ligands, such as the progesterone receptor, estrogen receptor, these motifs bind the ligand. They have a DNA binding domain, which is important for identifying the target gene in the promoter region and binding the promoter region of the gene. And then you also have regions that are important for regulation of transcription. Most nuclear receptors have an activation function in the ligand binding domain and also in the amino terminal of the receptor. You can actually conduct, and people have conducted a evolutionary hierarchy of nuclear receptors, and the ones we're interested in are over in the upper right-hand corner. We have the nuclear receptors: androgen receptor, progesterone receptor, glucocorticoid, and mineralocorticoid receptors, and these are highly related. In fact, the DNA binding sites they bind are very similar. More closely related to them is the estrogen receptor, ER alpha and beta, which is also important for the regulation of uterine function. And what I'll talk to you a little bit about today too is the more evolutionary primitive nuclear receptors, the chicken ovalbumin upstream promoter transcription factor 2, which is also involved in the regulation of endometrial function. The next slide shows the evolution of how science perceived nuclear receptor signaling. Prior to the 1970s, it was thought that steroid hormones worked by binding the membrane and allowing nutrient flux in cells, and that's how they regulated growth and function of the cells. Then in the 70s, with the advent of molecular biology, the hypothesis was, and what was proven to be true, is that nuclear receptors are transcription factors. They come in, they bind the DNA in the promoter region of a gene, they interact with RNA polymerase II, RNA is then generated in a gene-specific fashion, proteins are made, and the cell function is changed. So this is work that was done by Pierre Chambon, Bert O'Malley, and a whole bunch of people in the 70s. Then as the field evolved, they showed that the nuclear receptors don't act alone with RNA polymerase, but there are actually adaptive proteins that connect the nuclear receptors to the transcription factor machinery that allow for transcription. And then finally, in this millennium, it's shown that these steroid hormone receptors bind with co-regulators. These co-regulators not only function to work with the RNA machinery to make RNA, but they also modify the chromatin to allow more access of the transcription factors to these binding sites to ease the transcription of these genes. And I think one of the most advanced models is that the co-regulators, the ones that interact with the nuclear receptors, are diagrammed in B here, where they have protein-protein interaction domains in the amino terminal, and in the carboxy terminal, they have enzymatic activities that allow them to alter the DNA and the chromatin that facilitates transcription. So when these co-regulators bind the receptor, they can interact with other co-regulators that will modify the chromatin, they'll acetylate the chromatin, they'll open up the chromatin to allow access of the DNA to more proteins for RNA polymerase to come in and transcription to occur. So now when we look specifically at how progesterone receptor works, the initial hypothesis on how progesterone receptor work is that the receptor lives in the cytoplasm. And it's surrounded by chaperone proteins, heat shock proteins, and FKBPs, which keep it in the cytoplasm. When ligand binds, it frees it up from these chaperones. The receptor comes into the nucleus, binds its response elements, interacts with co-regulators, and regulates target gene transcription. In the late 80s and early 90s, a novel mechanism of progesterone receptor signaling was discovered by Bert O'Malley's group, where they showed that you could get activation of the receptor independent of hormone. Membrane receptors would, by peptide hormones, would activate these receptors. They would phosphorylate the progesterone receptor, which then would come into the nucleus in a ligand-independent way, and then regulate target gene transcription. So you could get activation of genes independent of the ligand. And then later in this century, Dean Edwards showed that not only did progesterone receptor act in the nucleus, but ligand to progesterone receptor could bind membrane receptors, activate their signal transduction cascade, and actually activate transcription factors independent of progesterone receptor to regulate gene transcription. So if you go back to the old 1970s, progesterone binds its receptor to regulate gene transcription. It is a little bit more complicated than that. So our goal over the years has been to use the mouse as a model to identify the role of progesterone receptor in vivo, to find out, what aspect of progesterone receptor signaling is governing uterine function. So now we're going to talk about defining the role of progesterone receptor in the mouse uterus. So again, progesterone receptor, we study it. It's by name. It's the progestational hormone. It's the hormone of pregnancy. And it's necessary in the uterus to regulate the window of receptivity. It regulates the ability of not only to support embryo attachment and invasion, but to support future embryo growth, and it regulates myometrial quiescence. Since uterine physiology is governed by steroid hormone signaling, most of the diseases of the uterus are associated with altered steroid hormone action. For instance, endometriosis, endometrioma, and endometriosis, endometrial cancer, and leiomyoma are all associated with altered progesterone receptor signaling. At least aspects of different of those diseases are. So this is the progesterone receptor. It's very similar to the stick figure we described of most nuclear receptors. It has a ligand binding domain. It has a DNA binding domain. It has an activation sequences in the ligand binding domain, and it also has activation sequences in the amino terminal to the DNA binding domain. The progesterone receptor has two isoforms: the shorter PRA isoform and the longer PRB isoform. The PRB isoform, these are encoded by the same gene, where promoter usage starts two different translational start sites. And the B codes for 166 amino acids longer, and it's thought to have an extra activation domain, and it's thought to be a more potent regulator of gene transcription, whereas the PRA is thought to be a less potent regulator of gene transcription, and in some cases actually an inhibitor of gene transcription. And this just summarizes what I've talked to you before, the three different pathways of progesterone signaling. You have the ligand-dependent pathway, the ligand-independent pathway, and the cytoplasmic pathway. So when we were initially going to go look at the progesterone receptor in a mouse, a lot of people said, well, why don't you just remove the ovaries, and then you remove ligand, and then you can study progesterone receptor action. And in that case, we would only be studying the middle pathway of progesterone receptor action. It wouldn't tell us what progesterone receptor was doing in a ligand-independent way, nor in the cytoplasmic action of progesterone receptor. So what we decided to do was to generate an animal model that had no progesterone receptor. And that would allow us to define what does progesterone receptor do, independent of the many pathways it could act, and then go back and try to piece back the molecular mechanisms by which progesterone receptor acts. So this is work that's actually 20 years old. The publication is now 20 years old in October, I believe. And John Lydon made a targeting construct where he would knock this into the progesterone receptor gene, where he would insert the neomycin resistance gene after the start of translation of the B and A isoform. This would knock out both isoforms of the progesterone receptor. We made mice with this construct. And then in the old days, we validated that progesterone receptor knockout was effective by actually doing ligand binding. So we took uterus, we ground it up, we added radioactive progesterone, and then determined the binding kinetics of it. You see the wild type is a nice saturation curve for binding of radioactive progesterone. The heterozygote, which is missing one copy of the progesterone receptor, has half the amount of progesterone binding, and the knockout had no progesterone binding. And this validated that we had knocked out the progesterone receptor in the mouse. Western blots showed that there was no protein being made. The next slide summarizes the overall phenotype of the progesterone receptor knockout mouse. What this told us is that progesterone receptor in the mouse was critical for every aspect of pregnancy. Female mice after estrus will undergo a stereotypic behavior where they arch their back and present their genitals to the male. It's called a lordosis response, and when you knock out the progesterone receptor, the females don't show the lordosis response. The females didn't even ovulate, so progesterone receptor regulated ovulation. We initially showed that these uteri had an increased estrogen sensitivity because they lacked the ability of progesterone to inhibit estrogen sensitivity in the uterus. The uterus couldn't undergo a decidual response, and there was disrupted mammary gland morphogenesis. So getting pregnant, maintaining pregnancy, and supporting the fetus after birth was all dependent upon the progesterone receptor. We then focused on studying the progesterone receptor in the mouse uterus. So this just shows the morphology of the mouse uterus at 3 different stages. Right after ovulation, prior to the implantation window, the pre-receptive phase. In the middle, we have the receptive phase, and then we have the post-implantation phase. The uterus has surrounded by a smooth muscle layer of the myometrium, surrounding the glandular endometrium, which has a luminal epithelium, a glandular epithelium, and fibroblastic stroma. Mucins keep the embryo from attaching to the epithelium at this stage. At the window of receptivity, the uterus closes around the embryo. The mouse can attach to the epithelium and initiate the decidual response in the stroma. These fibroblastic stromal cells increase in ploidy and DNA content, protein content, RNA content. They become epithelial-like, forming tight junctions which protect and nourish the embryo. And this is all coordinated in the mouse from the ovary. The next slide is a diagram of the pseudo-pregnant mouse starting from the estrous cycle going through pregnancy. At estrus we have the ovulatory surge of estrogen followed by a surge of progesterone, which then drops. And then as pregnancy progresses, in the pre-receptive phase, we have an increase in progesterone. Prior to the receptive phase, where the embryo can attach, we have an increase in estrogen, which triggers the implantation response. And then progesterone receptor is maintained by the ovary until the embryo can rescue the corpus luteum from dying by secreting placental lactogen. And this is similar to the human, except in the mouse where everything is contracted to 5 days in the receptive phase, things in the humans are spread out over a longer period of time, where we have our estrogen surge, our increase in progesterone after ovulation, during the luteal phase of the cycle, and then we have a peak of estrogen prior to the receptive phase to allow for embryo attachment. So things in the mouse are very parallel to the human, and this coordinates signaling in the mouse uterus. If we look at how the progesterone receptors change in the mouse uterus in the pre-receptive and receptive phase, we can see that in the pre-receptive phase, there's a little bit of progesterone receptors in the epithelial, the luminal epithelium glands, very little in the stroma. Prior to the window of receptivity, we have a peak of progesterone receptor in the epithelium. And then post-receptive phase, we lose progesterone receptor in the epithelium, and it switches to the stroma. So there is a time coordination of progesterone receptor expression in the mouse uterus during pregnancy, and it's very similar to what happens in the human endometrium during the menstrual cycle. We have progesterone receptor in the epithelium going away at the window of receptivity and switching to the stroma. The nice thing about the mouse is we can study decidualization independent of pregnancy. And this is what we use is a protocol that was developed by Finn and Martin in the 70s, where you overectomize mice. You mimic the ovulatory surge of estrogen by treating them with estrogen. You rest a couple of days. Then you treat them with progesterone and estrogen. Then you traumatize one uterine horn by either scratching the anti-mesometrial side or injecting it with oil. The traumatized horn will undergo a complete decidual response. The untraumatized horn will remain normal. You can actually weigh the decidualized horn versus the non-decidualized horn to get a quantitative assay of the steroid hormone responsiveness of the uterus. You can also look at morphologically at the stroma cells. They express alkaline phosphatase and change the morphology, and the traumatized horn, where they don't do that in the control horn. So when we knocked out the progesterone receptor, the embryos did not attach, and the uterus did not decidualize. You can see from the wild-type mouse we get a nice decidual response, and when we weigh the decidual horn versus the non-decidual horn, we can get a 20-fold increase in weight, whereas in the untraumatized horn we get no change in weight. So the embryos can't attach, they can't invade, the uterus can't decidualize, and if you treated the mice with estrogen, you can inhibit epithelial cell proliferation by treating them with progesterone, and in these mice you could not inhibit the epithelial-induced proliferation by treating with progesterone. Again, if we look at the mouse receptor, we have the A and B isoform as in the human. And initially, we knocked out the entire progesterone receptor by knocking out both A and B. Later on, Orla Keneally's lab mutated the translational start site for the A, or mutated the translational start site for the B, and then isolated the phenotype in mice that only made PRB or only made PRA. Where she knocked out the PRA and only made PRB, the PRKO mouse, they completely phenocopied the PRKO mouse. They were infertile, there was no embryo attachment and no decidualization. When she knocked out the PRB and only had the PRA, those mice were completely fertile and there was no discernible uterine phenotype. And this told us that in the mouse, the PRA is the predominant isoform, and if you look at expression levels, the mice makes a lot of PRA and very little PRB, which is unlike the humans, which tends to be a PRB-dominated uterus, or depending upon pregnancy, can switch from PRA to PRB. We also looked at the role in the global phenotype in the uterus of the steroid receptor co-regulators in the mouse uterus. So the steroid receptor co-regulators, the SRCs, there are 3 of them. The first one cloned was SRC1, then there was SRC2 and SRC3, and these are co-regulators. They have regions that on the amino terminal that interact with the receptors and other proteins, other co-regulators, and they have functional assays on the carboxy terminal that will modify the chromatin and allow for transcription. And again, this just shows that the co-regulators bind the nuclear receptors, they interact with other co-regulators to regulate transcription, and the SOC families interacts with transcription factors, with tumor suppressor genes, with other co-regulators and kinases to regulate tissue function. The first SOC to be knocked out in mice was SOC1 by Jae-Ming Hsu when he was a postdoc in Bert O'Malley's lab. And the mice were viable and fertile, but when you conducted a decidual response, you can see that the SOC1 mice did not respond, had a muted decidual response. We then looked at the decidual response in the SOC2 and then the SOC3. So the SOC2 was on the left, the SOC3 knockouts in the middle, and then this is a summary of SOC1, 2, and 3. And you can see that when you knock out SOC2, as with SOC1, you get a reduced decidual response, whereas when you knocked out SOC3, you had no phenotype, no effect of steroid hormone signaling in the uterus. So the next question is, does SOC1 and 2 compensate for each other? So when you have mice with both knockout of SOC1 and SOC2, they die. They're lethal. I mean, SOCs are expressed in multiple tissues, and when you knock them out, the mice are not born, don't even make it to birth, they die. You cannot study uterine phenotype. So what we did was we took the SOC1 mice, which were pretty hardy, and we knocked out SOC2 in those mice specifically in the uterus using our PRKO model. So we made mice that had their whole body SOC1 knockout and had an SOC2 knockout in progesterone receptor expressing cells. The PRKO is expressed in the pituitary, ovary, uterus, and mammary gland, but we focused on studying the uterus in these mice. And what you can see is that when we knocked out both SRC1 in the whole animal and SRC2 in the uterus, we lost the ability of the mouse to undergo a decidual response. The mice were sterile, there was no implantation. So it told us that progesterone receptor was key and that the level of SOC1 and SOC2 worked together to get full progesterone receptor signaling in the uterus. Work by Bert O'Malley's group has then gone on to look at the roles of these co-regulators in humans. And then, as we'll talk to later, you can collect primary fibroblasts from women, culture them in vitro. They will proliferate. You can treat them with a cocktail of hormones, which we'll discuss about later, and they'll undergo an in vitro decidual response. They change in morphology and gene expression. And from work done by Bert O'Malley's group, and this is a review by Maria Sfark and John Lydon, what they show is that one of SOC2's functions is to regulate carbohydrate metabolism. It regulates fructose metabolism. So when you knock out SOC2 in these stromal cells, they have a decreased proliferation, and they cannot metabolize carbohydrates. They don't have the engine to undergo a decidual response. Work by Sanjun Han in Dr. O'Malley's group has shown that SOC1 has a critical role in endometrial disease. So as you know, when women menstruate, sometimes the menstruation goes retrograde into the peritoneum. Most of the time, the cells that end up in the peritoneum undergo apoptosis and are forgotten about. But in some cases, these cells are maintained and they form endometriotic plaques, which cause pelvic pain and can be a source of infertility. What Sanjun Han showed was that in patients with endometriosis, the cells don't undergo apoptosis. The inflammation caused by the endometriosis will cause a processing of the SRCs to get a truncated version, which instead of promoting apoptosis, inhibits apoptosis and promotes proliferation. So you can see that these co-regulators are clinically relevant to identify what is going on in human endometrial disease. So now I want to talk about identification of pathways involved in progesterone receptor signaling. What does the receptor do? Okay, we knock it out. It causes uterine function to go wrong. What processes is it altering? And what we've done over the years was do DNA microarray. Now we're doing RNA-seq analysis where we can treat mice with vehicle of progesterone, both the wild type and the knockout, and ask what is affecting gene expression. As we'll describe later, we can identify by using chromatin immunoprecipitation followed by whole genome sequencing where progesterone receptor is binding in the genome. So we can identify genes that are affected by progesterone receptor and then identify their direct targets by which genes are bound by progesterone receptor. And I show this for historical purposes. This was our first attempt at identifying what genes are regulated by progesterone receptor in the mouse. We took wild type and PRKO mice. We treated them with progesterone. We isolated RNA and we probed the 1.1K atlas array blot. And you can see here we have one dot that's very low in the knockout and then dark in the wild type. And this turned out to be Indian hedgehog. This turned out to be a progesterone receptor target that's expressed in the mouse uterine epithelium. Indian hedgehog is a member of the hedgehog morphogen family. These are secreted growth factors that regulate tissue function. The members of the hedgehog morphogen family are Indian hedgehog, sonic hedgehog, and desert hedgehog. Sonic hedgehog is the most famous. You knock it out, it affects CNS, limb, multiple organ development. You knock out desert hedgehog, and it affects testicular development and germ cell development. Indian hedgehog is the only hedgehog expressed in the mouse uterus. We showed it was regulated by progesterone receptor. When you knock it out, half the mice die because they cannot form a vascular yolk sac, which says Indian hedgehog may regulate vascularization, which is part of pregnancy. And the other half die because they can't form a rib cage, and they suffocate at birth. And hedgehog is shown to have been altered in the expression of women with endometriosis. One of the genes that's been shown developmentally to be downstream of sonic hedgehog is the orphan nuclear receptor ChIP-TF2, chicken ovalbumin upstream promoted transcription factor. It's an orphan because nobody's identified a ligand. Its pattern of expression indicates a role in epithelial-mesenchymal communication. In the CNS, sonic hedgehog from the epithelium regulates ChIP in the stroma, which regulates central nervous system development. In mouse uterus, Indian hedgehog is expressed in the epithelium, and ChIP is expressed in the stroma. When you knock out ChIP, the mice die because they can't form a vascular system, which is nice when you look at implantation. This gene is so important that when you knock out one copy of ChIP, the mice have severely reduced fertility. In fact, they have a decidual defect. And ChIP-TF2 is actually lost in endometriosis in women. Sutter-Bullen showed that a long time ago. And they hypothesized that this was a critical regulator, inhibitor of aromatase, which is critical for endometriosis growth. So what we did was we made a PRKO, which we can knock out genes in the uterus. We crossed it to mice with conditional alleles, which allow them to be ablated by KRI for hedgehog and ChIP. We looked at ovulation in the hedgehog and ChIP mice, which was normal. We looked at fertilization, which was normal. We looked at progesterone levels, which was normal. So this told us that when we knock out hedgehog and ChIP with our PRKO, ovulation and the neuroendocrine axis was intact, but the mice were sterile. So we looked at the implantation and decidualization. And this is work by Kevin Lee, a grad student in my lab from a long time ago. And it follows the same for ChIP-TF2. When you knock out Indian hedgehog, you get no implantation. There's no implantation walls. There's no decidualization. on the artificial decidual response. In the wild type, the embryos attach. In the knockouts, the uterus does not zip around the embryos, and the embryos stay free floating in the uterine lumen. Why don't the embryos attach? Well, normally MUC1, a protein that the glycoprotein that prevents embryo attachment, is lost in the uterine epithelium, whereas in the hedgehog and coop knockouts, it's maintained. So what we were able to deduce by a lot of molecular biology, microarrays, and inhibitor studies was to show that an Indian hedgehog will bind its receptor from the epithelium, will bind its receptor in the endometrial stroma in the pre-implantation period. You'll get activation of this G protein smoothin, which will regulate COOP expression. It'll regulate epidermal growth factor signaling. The COOP will then produce growth factors, will regulate the expression of growth factors and inhibit them so that you don't get activation of estrogen receptor and increase in estrogen receptor signaling, and you don't get a decrease in you'll inhibit estrogen signaling, you'll get a decrease in MUC, and you'll allow for embryo attachment. So we have an epithelial to stroma back to epithelial reflex loop that allows for embryo attachment. We also showed that hedgehog expressed in the epithelium, when progesterone comes in, it activates hedgehog. Hedgehog goes to the stroma. You get activation of COOP. It allows for the process the embryos can attach. Then allows for these stromal cells to decidualize. It does this by activating members of the TGF-beta family, members of the Wnt signaling family, which regulate proliferation, survival, immune repression, and angiogenesis and decidualization. So having done the whole uterus, looking at the signal pathways, we then wanted to go and look at the role of epithelial, specifically what does the progesterone receptor do in the uterine epithelium. And again, receptors expressed in both epithelial and stromal compartments. You get a regulation of genes involved in growth factor crosstalk, and we want to identify what progesterone receptor was doing in the epithelium. This was critical in light of work done by Jerry Cunha and his collaborators and Paul Cook, looking at tissue reconstitution. Before you could do cell-specific knockouts, what they did was they could take isolate epithelium from mouse uteri from wild type, which had progesterone receptor, and from stroma, which had progesterone receptor, recombine this, put this in the kidney capsule, and say, what happens to Indian hedgehog expression? And when you give progesterone, you get induction of Indian hedgehog expression. Then they could ask, what happens if I give estrogen followed by progesterone? Do I get inhibition of proliferation? And they got inhibition of proliferation. They then took PRKO epithelium and PRKO stroma with no progesterone receptor, and they showed that when they recombined those, put those in the kidney capsule, they didn't get induction of gene expression, and they didn't get inhibition of proliferation. So then they mixed and matched wild type and knockout. So when they took wild type epithelium and knockout stroma, it phenocopied the PRKO, where we had no induction of target genes, no inhibition of cell proliferation. When they took stromal intact progesterone receptor and combined that with a knockout epithelium, they got a wild type phenotype. And what they concluded was that progesterone receptor in the stroma is the major driver of human function, of uterine function in these models. And we were kind of thinking that maybe what's going on in vivo. So Richard Behringer made a creed that would allow us to knock out genes specifically in the epithelium. And we made a conditional allele where we floxed the DNA binding domain of the progesterone receptor. And as you can see here, at day 2.5 and 3.5 of pregnancy, in wild-type mice, we get normal expression of progesterone receptor in the epithelial compartment and the beginning of expression in the stroma. In our Wnt7a Cree, we knocked out progesterone receptor expression in the epithelium, but maintained it in the stroma. And then in these epithelial-specific knockouts of the progesterone receptor, we can see it mimicked the PRKO. The embryos floated in the uterus, weren't able to attach or invade. There was no decidual response. And there was, in the mice, when you give estrogen, you get epithelial proliferation. You give progesterone and estrogen, you get inhibition of that proliferation. And in our epithelial-specific knockout, we were not able to inhibit that proliferation. So this told us that the epithelial progesterone receptor was really doing something important. And in fact, When we looked at target gene expression, the epithelial target genes, Indian hedgehog, GATA2, CYP26A1, and amphiregulin, the epithelial knockout phenocopied the PRKO. We got no induction in response to progesterone of these target genes. The stromal target gene, IL-13RA2, was not affected by epithelial, but was affected by the total PRKO knockout. In fact, we were able to show that progesterone receptor bound the Indian hedgehog gene close to the promoter region and 19 kb away from the promoter region, showing that the receptor bound hedgehog, regulated gene expression, and epithelial receptor was critical for uterine function. This told us that epithelial PR is critical for regulating gene expression. It regulates attachment. It inhibits estrogen-induced proliferation and decidualization. So what are direct targets of progesterone receptor? And what we've done next is conducted chromatin and immunoprecipitation followed by whole genome sequencing. So in this case, we isolate, we cross-link, we take mouse uteri, we treat it with formaldehyde, which binds the protein to the DNA. We isolate DNA and sonicate it. Then we get an antibody to progesterone receptor and immunoprecipitate all of the DNA sequences, fragments bound by progesterone receptor, and then we do next generation sequencing to identify where is progesterone receptor binding in the genome. And this is just designed. We treated mice with oil and progesterone, we immunoprecipitated, we fixed with formaldehyde, we immunoprecipitated with PR, and we sequenced and tried to find out what were direct targets of progesterone receptor action. What this told us was that progesterone receptor in vivo bound near the promoter region of its target genes, although its binding sites were spread throughout the genome. We looked at the motifs and showed that the major motif was what was known was the progesterone receptor response hormone, what was already identified by people in the 90s by band shifts and footprinting. We had some full progesterone receptor binding sites and mostly half sites. We then took these sequences and said, what can we learn that's new from this? This is not new. So we took our ChIP-seq data, mined it on top of our microarray data, and what we discovered is that progesterone receptor bound genes involved in the circadian rhythm in the uterus. It bound clock, NPAS, CRY, PER, all these genes that are shown to regulate the daily fluctuation in gene expression. In fact, we could also show that progesterone regulated the expression, not only bound these genes, but regulated their expression. So when you think about in the mouse with the timing of receptivity is critical for implantation, by progesterone governing the circadian rhythm of the mouse uterus allows all the uterine cells to be synchronized so implantation occurs in a finite window. What I'm going to finish up and talk about is the role of the progesterone receptor in the human endometrium. And this is work we just published in Endocrinology and Molecular Endocrinology. As I said before, we can recruit patients, consent patients, isolate their the clinicians or clinical collaborators can isolate the endometrium from their uterus. We can culture primary stromal cells. We treat them with vehicle that remain like fibroblasts. We treat them with a cocktail of estrogen, methoxyprogesterone acetate, and dibutyl cyclic AMP, and they will change their morphology to an epithelial-like phenotype. And we could actually conduct RNA-seq to find out what genes changed. Between undecidualized and decidualized, we identified 4,000 genes are altered between these two states. We can then knock down the progesterone receptor, and they don't decidualize, they don't change morphology, they don't express decidual markers, and we conduct RNA-seq and we can find out that about 5,000 genes are altered. And this just shows, again, the schema of how we did it. And this just shows that normal uterus, undecidualized. 6 days later, you see this cobblestone decidualization. In this case, we knocked down EGFR, and we could see the fibroblastic phenotype lacking the decidual morphology. The next slide just shows that when you knock down EGFR, you inhibit decidualization. The target genes are IGF binding protein 1, prolactin, Win4, are all targets of decidualization if you don't want to go on morphology. When we looked at our RNA-seq for progesterone receptor versus just what changes in decidualization, we find that during decidualization, some genes are not affected by progesterone receptor knockdown. And these are genes involved in glycan synthesis, glycosamine and glycan degradation, arginine proline metabolism, and some TGF-beta signaling. Then there's about 2,100 genes that are altered in decidualization and are altered by progesterone receptor knockdown, and those are signaling pathways such as P53, WNT, JAK-STATs, focal adhesion, pathways in cancer, and cell cycle and proliferation. And then the genes that are regulated by progesterone independent of decidualization are more of your metabolic glycogen metabolism enzymes. We then can conduct ChIP-seq. We can show that progesterone receptor binds at about 8,000 sites in the human genome. The major site is a progesterone response element. But we look at the other motifs that come up when we do this ChIP-seq, and we see that there's a Forkhead motif that comes up, a Hox motif comes up, an interferon regulatory factor motif, and a FOSL2 motif. So aside from progesterone receptor, maybe progesterone receptor is interacting with these other transcription factors in regulating decidualization. And the one we focused on, one that was done by pioneering work by Stuart Hanwergen and Jan Brozens and Julie Kim, is Foxo1. Foxo1 has already been shown to be a critical regulator of decidualization in human stromal cells. It's a member of the Forkhead transcription factor. It has a helix loop helix turn DNA binding domain, and it regulates a lot of cellular processes. It inhibits the cell cycle, it regulates oxidative stress resistance, it regulates apoptosis, and it regulates metabolism. So we repeated Stuart Hanwergen's work and Julie Kim and Jan Brozens' work, and showed when you knock down Foxo1, you inhibit decidualization. And then when we looked at the genes that were altered, a lot of the genes that were regulated by progesterone receptor were also regulated by Foxo1. Focal adhesion, P53 signaling, extracellular matrix. We then conducted ChIP-seq, and if you remember, progesterone receptor only bound 8,000 sites, Foxo1 bound 26,000 sites all throughout the genome. The major binding motif was a Forkhead motif, but we also had June motifs, Runx motifs, SOX motifs, and PR motifs. When we overlaid these 26,000 sites with the 8,000 sites from progesterone receptor, you can see that 2/3 of the progesterone receptor binding sites are also occupied by Foxo1 binding sites, which to me is pretty phenomenal. So the question we asked was, does Foxo1 regulate the ability of progesterone receptor to bind these sites? So we knocked down Foxo1. It did not affect progesterone receptor A or B expression in the stromal cells. But we looked at, could progesterone receptor bind target genes, IGF-1, CDK1, ZEB1, and KLF15? You can see that in normal wild-type cells, progesterone receptor bound all of these binding sites in the promoter region of these genes. However, when we knocked out Foxo1, progesterone receptor, even though the protein level is expressed normally, could not bind its target site. And if it could bind it, its binding is reduced. So we came up with the model that normally PR and Foxo1 opens up the DNA for progesterone receptor to bind. If you don't have Foxo1, the chromatin can't open, progesterone receptor can't bind. So what to summarize this entire talk, we've utilized genetically engineered mouse models combined with cystromics and transcriptomics to identify pathways that regulate uterine function in the mouse. PR, along with its co-regulators, act in the uterine epithelium to establish a crosstalk between the epithelium and stroma to allow for attachment and stromal decidualization. Using primary stromal cells, we've shown a role for progesterone receptor in the interactions in human decidualization. I mean, this work is done by a lot of collaborators, creative grad students, postdocs, and clinical collaborators. I'd like to thank Urcha Gavachi, Bill Gibbons for their help on this. This work was funded by NICHD and the U54 SIPR program. I can now be open for questions.
Dr. DeMayo?
Yes, sir.
Question for you. This is Angela Barbara. Got my audio working now. I have a question for you. Why do selective estrogen receptor modulators have different effects in different estrogen responsive tissues? Or why do the same question would apply to progesterone receptors?
And that's because when the ligand binds, it alters the conformation of the receptor, and it alters its interaction with co-regulators. And the composition of co-regulators is different in different tissues. So in the case of tamoxifen, it may be a total antagonist in one tissue and a partial agonist in another tissue, because the conformation of the receptor allows it to interact with co-regulators or co-repressors differently.
Okay, so then as a follow-up question, can one confer progesterone responsiveness, for example, on a tissue that does not normally have progesterone receptors by artificially expressing progesterone receptors in that tissue? In other words, do non-progesterone responsive tissues have the post-receptor signaling pathways that work with progesterone receptors? Or do, for example, steroid responsive tissues have to have the entire cascade from receptor to co-regulators?
So the expression pattern of the co-regulator is not as restricted as the nuclear receptor. So there's a lot of tissues, liver, brain, muscle, that have the co-regulators. I think right now we've generated mouse tools that will allow for the expression of progesterone receptor and other nuclear receptors in other tissues. And I think you cannot convert a muscle to a uterus response just by expressing progesterone receptor, because remember the chromatin is epigenomically modified differently. So the genes that are available for regulation by nuclear receptors are going to be different in muscle and uterus. I'll give you an example of some unpublished work we have where SF1 is a nuclear receptor that regulates aromatase. And it's not expressed in normal endometrium, but it's expressed in endometriosis. And it's thought that it regulates aromatase and it causes the endometrial cells to live outside independent of estrogen. We made mice that we expressed SF1. And the mice gave a cystic uterine phenotype, which was reflective of endometriosis. However, the gene expression pattern was different because some genes are methylated or shut off, and no matter how hard you express the nuclear receptor, if it doesn't have those pioneer transcription factors and co-regulators, you're not going to get the same. So what I would say is if you express the receptor in non-receptor tissues, you'll get some target genes to be altered, but not all. And this is true when you look at endometrial cancer cells. A lot of endometrial cancer cells, like HEC1A cells or Ishikawa cells, you can engineer them to express the progesterone receptor, but they don't quite mimic the in vivo situation because the epigenetics of the chromatin is different, as well as the cohort of co-regulators. So you need both factors to get full responsiveness.
Excellent, thank you. I'd like to thank Dr. DeMayo for a really excellent and very lucid presentation about steroid hormone action, and just let you know that Dr. DeMayo is going to be in the very near future relocating from Baylor College of Medicine to the National Institute of Environmental Health Sciences. The next webinar in our series will take place on Tuesday, July 21st at 10 a.m. Eastern Time, and it will be given by Dr. James Liu of Case Western Reserve University, who will be speaking about clinical pharmacology. Thank you very much, everyone, for your participation, and we apologize for the technical glitches that we had today. Thank you, Franco.
Take care, Andy. Thank you. Have a happy 4th of July.
You too.